part 2 of the tutorial on the molecular biology of inheritance. And remember, this is just going to be just a quick little kind of like a helper video, maybe to help you understand a little bit about this chapter. And we got to the part where we were talking about DNA being replicated. Now we need to talk about the next part that I think is probably the most confusing to students, is the process of transcription and translation. If I ask you to copy a passage out of an encyclopedia, I'm asking you to transcribe that. So transcription is basically the process of copying or making a copy of. Translation in like an everyday text would mean that you're going to take something that I give you and you're going to turn it into another form. And that's kind of where we are with this this next idea. Transcription takes the DNA that you have in the nucleus. Now the DNA can't leave the nucleus. So somehow those instructions that are in the DNA have to get out to the ribosome, and they do that through RNA. DNA is transcribed. Transcribe, remember, meaning to take something and copy it onto RNA. The process of translation, you're going to take that RNA copy and you're going to turn it into, you're going to translate it into an amino acid, a protein. Alright, so basically here's how it works. We go from DNA, this process from going to DNA to RNA is called transcription. So we go from here to here, that's transcription. When you go from RNA to a polypeptide to a protein, that's called translation. This word codon you're going to see come up over and over again. That's just a 3-base sequence like C, G, T. That would be a codon. And this is our codon chart, how we would read that. Now let's just say if I gave you the DNA strand here. What if the DNA strand was A, T, C, C, G, T? And I asked you to give me the other side of the DNA strand. The other side of the DNA strand would be T, A, G, G, C, A. What if I then told you to take this new strand and turn it into RNA? To mRNA specifically, and we'll talk about that in a minute. T binds with A binds with, not T, U. Remember, RNA has uracil. G binds with C, G, C, C to G, and then A, not T, it's U. So this would be our RNA strand. Now how do we go from RNA to a protein? Let's divide this up into codons. A, U, C. First thing you're going to do is on this side find A. It says our first base is on this side. So we know because the first letter is A, it's going to be in this orange block, in these rows. The second base is up at the top. Our second base is U. So I'm going to find U. Now I know that between A and U, it's going to be one of these. The third base is this third column. Our third letter is C. So I'm going to find C, and when I go over, I know for a fact that AUC codes for isoleucine. Isoleucine, excuse me. Let's do the next one. CGU. I'm going to find C, so I know it's in this purple. G, right here. So now I know it's one of these. And the last letter is U, so I know it's that one. So it's going to be arginine. So that's how I use this chart. First letter, second letter of the codon, third letter of the codon.
At the beginning of transcription, the DNA containing the gene of interest unwinds, forming a template strand and a non-template strand. Only the template strand is used to synthesize RNA. An enzyme called RNA polymerase adds complementary RNA nucleotides to the newly forming RNA molecule. When the RNA polymerase reaches a DNA region called a termination signal, it releases the DNA strand and the RNA molecule.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
And you're going to find that some of this DNA is not used. It's hard to kind of see this diagram, but this diagram's in your book. You got these exons and introns. The exons are the important bits. The introns are not. So you got this thing called a spliceosome that comes in here and cuts out the introns, leaving you the exons. Then it's going to put a cap on it, a poly-A tail. This is called the processing before it leaves the nucleus. Now this, now that it's been processed, can leave the nucleus. This is our raw strand up here. This can't leave the nucleus yet. But then we put the cap in the tail and we take out the introns, and now this completed piece of mRNA can leave the nucleus. You've got tRNA. tRNA stands for transfer RNA. It's going to bring the amino acids to the ribosomes, and on the tRNA there's a thing called an anticodon. Now an anticodon matches up with the codons, and I'll show you on this diagram. Here is our tRNA. It's called kind of a hairpin structure. This anticodon in down here matches the codon on the mRNA. Here is our location where our amino acid is. So this tRNA brings this amino acid to the ribosome. Let's look at that.
Translation is the final stage of gene expression. It occurs at the ribosomes in a cell's cytoplasm. During translation, amino acids line up in the order specified by an organism's genes. The amino acids are joined to make chains. These chains are then made into proteins. Translation is often called protein synthesis. Translation begins when a messenger RNA molecule binds to the ribosomal RNA in a small ribosomal unit.
Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid. Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the 3-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds of proteins every second.